name is Lynn Markey, and I'm here on behalf of the Coalition for Life Sciences. I want to thank you all for joining us today for our very last briefing of the 2009 Congressional Biomedical Research Caucus briefing. Today we have Dr. Stephen Warren with us. He is the William Patterson Tinney Professor of Human Genetics and Professor of Biochemistry, Pediatrics, and Genetics at Emory University School of Medicine. He received his PhD in Human Genetics from Michigan State University. Prior to joining the faculty at Emory, he did postdoctoral research at the University of Illinois School of Medicine in Chicago and was a visiting scientist at the European Molecular Biology Laboratory in Heidelberg. Dr. Warren is a diplomat of the American Board of the Medical Genetics with subspecialty certification in both clinical cytogenetics and clinical molecular genetics. He is a founding member of the Fellow for the American College of Medical Genetics and is a member of the Human Genome Organization. Dr. Warren has held various committee memberships in the Human Society of Human Genetics and was elected to the Board of Directors in 1997. Among his awards are the William Rosen Research Award of the National Fragile X Foundation and the William Allen Award of the American Society of Human Genetics. In 2004, Dr. Warren was elected to the Institute of Medicine of the National Academies, considered one of the highest honors in the field of medicine and health. So with that, I'd like to welcome Dr. Warren to the platform.
Thank you. Thank you, Lynn. I appreciate the invitation. I appreciate you all coming. I know this is a busy week for you all, and so I hope this will be useful to you. So what I'm going to tell you is a story about Fragile X syndrome, and then try to use it as lessons learned that may be applied to autism in the future. I have to make a disclosure. I'm chair of the Scientific Advisory Board of Seaside Therapeutics. So this is a slide we used to show probably now 20-some years ago when we first started the idea of thinking about cloning a gene that might cause a disease. And the idea was, if we could identify the gene, that gives us a toehold into the problem that then you can kind of understand what the mutation studies are in the family. You can do biochemistry. You can build models of that disease in either the mouse or the fruit fly. That allows you then to do cell biology and try to put together what the etiology of the disease is. And then once you get to that point, you can start thinking about therapeutics, because now you should have a thorough enough understanding of the process of that disorder that you can think about that in an intelligent way. And I hope by the end of this, I'll convince you that for at least Fragile X syndrome, we've pretty much closed this circle right now. That doesn't look good, does it? There's supposed to be a picture of a boy with Fragile X syndrome. But what you would see if that had worked, which is surprising, it almost always works, is that these patients with Fragile X syndrome really don't look like, say, Down syndrome. They're not very identifiable by most people. They look pretty normal. They have very slight features that are in common among patients. Their ears tend to be a little larger than normal, but not to the point of being unusual. So most of these children, and most of them are boys, are diagnosed about 36 months of age when they start not meeting their developmental milestones, usually a delay in language, and then they get tested for the disorder. The biggest impact on the patient is the intellectual disability. So these patients in general have an IQ that is about 40. And they also have many features that are shared with autism. So they have gaze avoidance. They don't necessarily like to be touched. They have, many of the features, and in fact, so many that about half of the patients now meet all the criteria for autism and can be diagnosed with that criteria. If you go into an autistic population, usually about 2 or 3% of those kids turn out to have Fragile X syndrome. So anyway, you can see that this young man is a pretty good-looking boy. But there is a resemblance, and here you can see, for example, in 4 boys of different ethnicities, they tend to share kind of a facies, but nothing's really abnormal. So the Fragile X syndrome gets its name because if you look at chromosomes under the microscope from these patients, the X chromosome looks as if the tip of it is going to break off. And this was noted back in the 1960s and became known as first the marker X and then finally the Fragile X, and then the syndrome was named Fragile X syndrome. It's formally called an X-linked dominant with reduced penetrance. And what that means is being on the X chromosome, it affects boys more than girls, but girls can be affected. About half of them have an intellectual disability. The prevalence of Fragile X syndrome, we just finished a large newborn screening in Georgia and found that the prevalence is about 1 in 5,000, which is not too far off what we thought it had been for many years. So compared to other things we, for example, screen for in newborns, like galactosemia is 1 in 50,000. So you can see this is a fairly common genetic disorder. The gene itself that causes Fragile X syndrome resides right at that gap on the X chromosome that we see under the microscope. And it is called FMR1. And this gene, what was unusual about it when we first identified it is there was a repeat within the beginning part of the gene, and it's a CGG repeat. And if you then look at this in normal individuals, there's about 30 copies in general of this CGG repeat. But if you look at patients with Fragile X syndrome, they almost always have greater than 200 copies. The average is about 800 copies. So it undergoes a massive expansion when it gets transmitted down through a family. And this was the first disorder identified with these trinucleotide repeat disorders. And you may have known, like Huntington's disease was found a couple of years later to have a similar kind of mutation, a myotonic dystrophy. There's all told now about 20 different human disorders that have this kind of mutation, but Fragile X was the first of those. When the gene does get to that threshold of about 200 repeats, something happens, and this is still one of the black boxes with Fragile X, even though we understand a great deal about the disorder. It turns the gene off. So what happens is, this represents the DNA here, and these little hash marks are all positions within the DNA where a base can be modified, and it's called methylation. And when that repeat length gets above 200, this gene gets heavily methylated and essentially turns it off. So it doesn't make any mRNA, and consequently there's no protein made. So really it's the absence of the protein that's called FMRP that causes Fragile X syndrome. So the repeat expands, it turns the gene off, no mRNA and no protein. Which that says. So consequently, a lot of research in the last 18 or so years has been focused on what does this protein normally do, because then we might be able to understand better what the consequence of its absence is in the brain. And we now know a great deal about what this protein does. We know it's a protein that interacts with RNA, with messenger RNA, and the messenger RNA, of course, is transcribed from the DNA, and then the messenger RNA is read by ribosomes to create proteins. And it's those mRNAs that this protein regulates. It's that protein synthesis step that it regulates, and it regulates only a handful of mRNAs. And it does so in certain locations in the brain, and that leads to the disorder. So a little primer on protein synthesis, like I just mentioned. So the mRNA is produced from the DNA, and then that mRNA is then read by the ribosome, these large bodies here, that then bring amino acids to each location and build a string of amino acids that ultimately create the protein that's encoded for by the mRNA and originally by the DNA itself. And then when you get many ribosomes on a stretch of messenger RNA, that's called a polyribosome, and I'll point that out to you in a second, what that looks like in the laboratory. So we know now a number of these messages that FMRP interacts with normally, and these are all messages that are unusual in some degree. And the unusual part of that is they tend to be located at distant locations in the neuron. So you all know neurons. You've seen pictures of neurons. You have the cell, and then they have these projections that go and connect to other neurons in the brain. It's at that faraway location at the tip of those, what we call dendrites, is where FMRP operates and where these messages are located. And it turns out that's where FMRP exerts its regulation of the production of protein in these distant locations. And so this is how we look at these polyribosomes in a laboratory. So this is simply a tube of sugar that we then put the cell, we lyse the brain cells, put them in the sugar, and we spin it real fast. And what happens is these ribosomes will go through this gradient depending on how heavy they are. So you have this big peak here is the monosome. So that means that's all the messages with just one ribosome. And then you have, this is with 2 ribosomes, 3 ribosomes, 4 ribosomes, 5, and so on. So these are polyribosomes here. Now, if we look at one of these messages that we know interacts with the FMRP, for example, this one encodes for a protein called MAP1B, we can see that normally in a wild-type animal, so now this is using a knockout mouse model, Fragile X syndrome, that about half of the mRNA is not in the polyribosome, so it's not being translated into new protein. But in a knockout animal, which is equivalent of Fragile X syndrome, almost all of that mRNA is in the polyribosomes, which means it's actively being translated. And indeed, if we look now at this protein itself in the brain of the knockout mice or the wild-type mice, you can see that there's more protein present in the knockout animal. So what is happening there is that FMRP is a regulator of translation or protein synthesis in the brain, and if you don't have FMRP, like in Fragile X syndrome, you over-translate or overproduce these proteins. So you lose the regulation, you make too much of these certain proteins that are very important with how neurons connect with each other. And this is just some data showing that, in fact, this FMRP is present in these. This is the synapse here, where one projection from one neuron is touching another projection of another neuron, and that's how they communicate, is across the synapse. And these little dots here are the FMR protein as seen under an electron microscope. It's important that it's there because if we look at the same sort of thing with a fluorescent microscope, looking at these dendrites in red, in green is this axon, you can see that unlike the wild-type mice, the knockout mice have these very long spines, and that's also seen in the brains of patients with Fragile X syndrome. And so somehow by misregulating the local protein synthesis, these dendritic spines have an unusual shape, and they actually have a shape of an immature spine in a dendrite. So basically up until this point, what we know then is that the FMR protein regulates local protein synthesis at these distant projections. And that's really quite critical because people like Eric Kandel, who won a Nobel Prize for his studies, actually showed that local protein synthesis is required for learning and memory. So if you don't allow the brain to make new protein, that brain's not going to learn anything because it needs to make new protein to lay down a memory, to learn. And so FMRP then appears to play a critical role in that ability to learn. And I know there's a real busy slide, but I want to just emphasize how much we now know about kind of what's going on in these distant locations with FMRP. And we know that there's a signaling pathway, and so this is the dendritic side of a synapse, so the axonal side would be up here, and then the axon will release glutamate, which is a neurotransmitter, and that will then interact with a receptor called mGlu-R, and then through a series of cascades of other proteins, ultimately it controls whether FMRP allows the message it interacts with to be translated or not. And if you don't have FMRP, then you lose all that regulation. And that's critical because we know the proteins that FMRP regulates also control another receptor in the same part of the synapse that controls whether this receptor is on the surface or whether it's internalized. And this is called the AMPA receptor. And the thing that's important about the AMPA receptor is the more AMPA receptor that's on the surface, the stronger is the connection between the two neurons. The more AMPA receptor is internalized, the weaker that connection is. So the amount of AMPA receptor really controls how you strengthen a synapse. And so obviously that's an important thing when you're learning something or you're trying to learn how to speak and language and things like that. You have to be able to rapidly acquire and strengthen synapse connections. And the ability to move AMPA receptors up onto the surface to strengthen that connection is critical to be able to do that. So this is experiments then we've done in the lab looking at this AMPA receptor. So I'll just walk you through. These are rat neurons in culture. And in the white panel here is the FMR protein. In the red is the AMPA receptor, but red is all on the surface of the neuron, and green is internalized AMPA receptor. So in a steady state, in a normal animal, you see most of the AMPA receptors on the surface. So it's kind of in the mode of learning and acquiring information. Now, if you reduce the amount of FMRP in a neuron, so now all the FMRP that's out in these distant locations is gone, you can see now almost all of the AMPA receptor has now been internalized. It's no longer on the surface. So by removing FMRP, like what happens in Fragile X syndrome, you have more of this AMPA receptor gets internalized, and that just results then of weakened synapse. So there's nothing inherently wrong with the synapse. All the components are there for making a memory. It's just weaker. So in a way, that's good news. I mean, if the synapse was broken, then I think it would be a lot harder to think about therapeutic interventions. But now it's just simply a matter of degree of strength of the synapse that appears to be the problem. So again, if you lose FMRP, you overproduce these certain proteins, and that results in excess internalization, and that's kind of the end game. That results in weakened synaptic strength, and that probably then is the reason why children with Fragile X syndrome have the intellectual disability and why they have the certain behavioral problems. It's simply this AMPA receptor. So I think over the past, it's been now 18 years since we found the gene. I think over that period, our lab and many, many labs around the world have learned enough that we kind of know precisely now why these kids have these problems. And it's simply the AMPA receptor on the surface is strengthening and weakening the synapse. And in fact, this is probably the best understood psychiatric or neurological disease there is. I think if you ask Tom Insel, the director of NIMH, he would say that Fragile X syndrome, we have so much detail, we've got down to so much granularity that we know precisely why these kids can't learn. And I think you can't say that about schizophrenia, bipolar, a lot of other disorders where we just don't know enough about it, autism. So knowing that has then allowed one to think rationally about maybe how one could intervene. And we had kind of serendipitously run into another neuroscientist, Mark Baer, who was then at Brown University, now at MIT. And Mark didn't really know much about Fragile X syndrome. He was working on how the eyes acquire vision and send that to the brain. But he was looking at a phenomena that occurs at the synapse, and I heard him talk at a meeting, and it kind of resonated with what we thought might be happening in these mice. And so Mark started working on Fragile X, and we were collaborating with him. But the end result is we came up with this theory that how one might pharmaceutically interrupt that problem and maybe correct it in some degree. And the idea is, so like I've been telling you, is that if you lose the FMR protein, you over-translate. And the signal for over-translating is that glutamate tickling the synapse. So when the axon fires and it stimulates this mGlu-R receptor, that stimulates translation to occur. And without Fragile X syndrome, that appears to be overreactive. I mean, it looks as if there's too much glutamate, but there really isn't. It's just reacting that way. So the thought is if you could dampen this signal here, maybe you could kind of ease up on the problem. So in some ways you can think of FMRP as the brake in the car and mGlu-R as the accelerator. And so if you lose the brake, you're going faster and faster and faster. But if you put a governor or something on the engine to slow everything down a little bit, maybe they'll bring things much more in balance. So that was the idea. One way you can test that is look at this AMPA receptor problem. So again, this is if you have FMRP in these dendrites, you can see most of the AMPA receptors on the surface. There's very little green, internalized AMPA receptor. If you lower the FMRP in these dendrites, you can see almost all the AMPA receptor goes internalized. But then if you add one of these drugs, which is called a mGlu-R antagonist, you can see that you can now correct that problem. So now there's still no FMRP, but now almost a great deal now of the AMPA receptor is back on the surface. So the idea is you can kind of trick the cell into thinking that it's not being stimulated, overstimulated, that you're backing it off. So then the AMPA receptor goes on the surface. Now this has been replicated in a lot of animal studies now in fruit flies. So we can make models of Fragile X syndrome in fruit flies. We can make models of Fragile X syndrome in a mouse. We can make models in a zebrafish. And all of these models, if you remove this protein, they have behaviors that either can be interpreted as learning and memory problems or behavioral problems. So in many ways, we think these models mimic the human disorder. And now a number of laboratories have shown that by using these sort of drugs, you can rescue all those phenotypes. So that's one class of drug, which is the mGlu-R antagonist. Then the other side of the coin is we also did a drug screen by using the fruit fly model, and we screened 2,000 drugs that include all the FDA-approved drugs and things like that, and just simply asked, do any of these drugs rescue the phenotype in the fruit fly? And in fact, we found several that did, and the most important class of drugs that we found were the GABA antagonists. And the reason why GABA is important, GABA is another neurotransmitter, but what GABA does is GABA is the opposite of glutamate. So glutamate normally stimulates the neuron, GABA normally inhibits the neuron. So it made perfect sense. So just like adding this, the antagonist to kind of weaken the stimulation from the accelerator, if you just kind of inhibit the entire neuron, it accomplishes sort of the same thing. So it was kind of made perfect sense when we kind of looked at the results and saw that the GABA agonists, in fact, were rescuing this phenotype. So the idea now is that we have two classes of drugs. We have the mGlu-R antagonist and the GABA agonist that can hopefully accomplish the same thing, and that is to reduce this overtranslation and bring more of the AMPA receptor to the surface and prevent the weakening of the synapse. And so the good news is that is now happening. So these are all the Fragile X syndrome clinics in the country that are part now of a consortium. So they're kind of trying to decide on common ways to evaluate the patient so that when clinical trials are underway, that we can do multi-center trials in a way that we can kind of pool the data together. And in fact, now there are all the centers in blue now have active Fragile X drug trials now underway for 3 different drugs that are, 2 of these are mGlu-R antagonists and 1 is a GABA agonist. And so those have just started, and we hope that there'll be at least 2 more drug trials, 2 different versions of kind of these drugs that'll be starting in the next year or so. So for Fragile X, I think hopefully you can see that we went around this loop in the last 18 or 19 years, and the toehold was really to get the gene. Before we had the gene, we had just absolutely no idea what caused Fragile X syndrome. We were very much in the same boat as we are with autism. We just really don't know what causes autism. We don't know how to separate patients with autism apart. We don't, because we know there's probably more than one disease, but we don't have the ability to do that. So that's really what we need to do with autism. And we think that the lessons learned from Fragile X will be very important. And the reason for that is that in this location in the dendrite where FMRP operates, in rare cases of autism, where there are known mutations in a certain genes, and this really accounts for very small numbers of autism, less than 1%, but in those cases where we know what gene is mutated and causes autism, those proteins also reside in the same place in the neuron, in the dendrite. So we think the kind of the biology and the biochemistry that we worked out with Fragile X syndrome will be applicable to autism, because it all looks like this same location that's abnormal in Fragile X syndrome is probably where things are going awry in autism as well. So what do we need to do? Well, I think we need to, as we do with Fragile X, we need to understand what the causes of autism is, or what a lot of people call it autisms, because it is going to be more than one disorder, it's going to be more than one gene. I think we need to be agnostic about what the etiology is. I think clearly there's genetic components, but it probably certainly is not all genetic. There's probably going to be environmental components. There's probably going to be epigenetic components, which are ways in which the environment can modulate the gene activity. And it may be something else we don't know anything about. I mean, we didn't know about trinucleotide repeat expansion prior to Fragile X syndrome. Maybe autism will be something else we don't understand. So I think we have to be able to fund kind of out of the box, think over the horizon with autism to be able to get a toehold. All we need is a toehold. Once you get a couple genes or a mechanism, then you can do that biochemistry. You can make those models, you can make the mice, you can make the flies. You can kind of do the basic science research that's required. And once you get enough knowledge, then you can start thinking about therapeutics. I think 5 or 6 years ago, if you asked me if there would be a drug for Fragile X syndrome, I would think probably not in my lifetime. I mean, outside of gene therapy, which really is not at all at the point of thinking about gene therapy for neurological or neuropsychiatric disorder, short of that, I mean, I couldn't think of a way you could possibly think of a drug. But, you learn enough, and all of a sudden it becomes kind of clear how you could pinpoint a drug to target where the problem is. And so we need that toehold. We need that gene or whatever the mechanism might be in autism, and that'll give us enough tools then to really investigate in detail what's going on in autism. So I think that's a big challenge now for people that work in this area of science. So I just want to thank all the people that fund my lab, of course, NIH and CDC, the Simons Foundation, and FRAXA. So thank you very much, and I'd be glad to answer questions.
Thanks, Dr. Warren, and that was a great speech. And typically at this point we do question and answer. I'd like to turn it over to Congressman Greg Harper if he has a few remarks.
And thank you, Dr. Warren, and some great information in there. I'm Greg Harper from Mississippi. I'm a freshman congressman. That just means I have all the confidence that goes with ignorance after 9 months trying to figure it out. But I have the distinction of being the only member of Congress who has a child with Fragile X syndrome. And so this is very near and dear to my heart, and I certainly would like to compliment the work that Emory University has done. And it's been an incredible deal. I just happen to have a picture of myself. So yeah, there you go. Pass it around there. So that's what I'm here for, right? But I wanted to just tell you a couple of things. I've got Scott Malvaney over there as my policy director, who's really taken the lead role in my office to make sure that we stay on top of this, and we are committed to make sure there's proper funding for the research that needs to be done on this and many other conditions. And we're also committed to some other issues. As you know, when you're dealing with children with special needs, when you get to secondary education, you drop off the planet after you leave high school. So we have, we're glad to report that we're continuing to work on that. You know, the 2008 Higher Education Opportunity Act talked about programs, demonstration models for children with intellectual disabilities, but as is the custom up here, there was no funding. So it was authorized but not appropriated. So we are hopeful at this point that funding will be there this time around. And we do know that Senator Thad Cochran from Mississippi took the lead on the Senate side and secured $14 million. We're hoping that will be there for that. So Do I have just a minute or two? Okay. My son, as that picture is floating around here, Livingston is our son, and he just turned 20. He was born in 1989. He was developmentally delayed, as is custom. He did everything at the tail end of the normal range. If it said you were supposed to do it between 9 months and 12 months, well, he did it at the almost the last day of the 12th month. But Fragile X syndrome has, we got a incorrect diagnosis originally. That we took him everywhere. When we were about 18 months old, the pediatrician finally said, something's wrong, let's start looking for it. And so we had an incorrect diagnosis of mild cerebral palsy and a near miss on autism. And that's what we lived with. But God does direct our paths. And our next door neighbor at the time was the head of all special education for our county public school system, adoptive aunt. And in 1993, when he was almost 4, she goes to their big education seminar they have every year in Mississippi. Just happens to go to a breakout session called Educating Children with Fragile X Syndrome. Had no idea what it was, went in, her mouth falls open, comes home and says, I think this is it. And sure enough, it was. He is fully methylated. But he is, the literature is very depressing on Fragile X. And so we threw the literature away. And so we have made Livingston live in the normal world. And so we have had him in inclusion classes through high school. Last year, he took classes at the local community college, took 2 classes each semester, made 2 C's and 2 B's, taking the same tests the other kids were taking. He just had to be read the tests, read the answers, and would tell them the answers. So they can learn. So he is adamant that he's going to go to college at Mississippi State University next year, so we're hoping that he may have that opportunity. He works 2 nights a week at a local restaurant, and he is a greeter at our church, and he is a great kid. If you took him, if you came in the room, you'd want to take him home with you. But, he is the face of why we do this. And so I want to thank you all for y'all's involvement and your support, and we're just going to keep working together and do what we can and keep praying for each other. Thank you very much.
Dr. Warren, I'll start the question and answer period. I'm wondering if there's a specific time in the development of a child that the drugs would be more beneficial than at other times. So if you have a 20-year-old versus a 4-year-old.
Yeah, so we don't know that answer precisely. But, I think obviously if you take someone who's 20, 30 years old with Fragile X who's, has not had the experiences or the learning throughout that period of time, you're not going to have a drug that immediately corrects everything. So I think it's going to be clear that the earlier one intervenes, the better. And that's why we did this newborn screen assay, so we now have a way to do that. So we could do newborn screening or infant screening to identify all the children with Fragile X syndrome so one could intervene early. It's probably unlike metabolic diseases that we screen for in newborns because you don't need to necessarily intervene at birth. This whole business of the synapse that's going on in the brain really doesn't start kicking in humans until 6 months of age, 7 months of age. So I think there's a window where if you could intervene, you might be able to correct it completely. But that being said, David Nelson at Baylor has made mice where he can turn the gene back on. So he can make a mouse that is born without this protein, like Fragile X syndrome. It grows into adulthood without the protein, but then he can turn the gene on. And those mice also reverse and become much, much better. So I think, I think we just have to keep an open mind, but it may not achieve the same degree of effectiveness in an older individual, but there might be some benefit. I would expect there might be.
Can you explain what the practical effects of this medication does? What changes? And then I'm just curious if Tourette's syndrome has any connection with Fragile X.
No, we don't know of a real connection with Tourette's. Well, these drugs, we know that a lot of them were developed initially for anxiety, but didn't really do a lot for anxiety, and that's why a lot of them were kind of shelved by the pharmaceutical companies until we kind of needed these antagonists, and they, a number of companies said, oh, we have some that we just never have done much with. We know they're pretty safe, that there isn't a lot of known side effects. So we'll just see how effective they are.
But do they make the patient learn, or do they allow them to learn better?
So that's what the trials are going to tell us.
Okay.
We don't know that.
The experiment where the 2 mice on one side were calm and the other 2 going crazy, what was that drug you gave them?
So that was called AMPA.
Oh, AMPA.
Yeah, but that's not a drug that has characteristics that would work good in humans. So, but these other drugs that are in trials are kind of just variants on that theme.
Interesting. Can I ask one
Sure.
more? You mentioned that there has been one autism gene isolated in the same area
Three,
as
actually.
the FMR1. Can you tell me the names of those autism genes?
Yeah, so one's called P10, for
P
example.
what?
P10.
P10?
Yeah. Then there's another gene called NF1.
NF01?
NF1.
NF.
And so anyway, there's those two of them.
Yeah, okay.
But I think all of the proteins that we know about in that whole location are all candidates for autism. So I think anywhere in that entire pathway, if something goes wrong, I think you probably, the end result could be something like autism. And we know, actually, because of advances in biochemistry and mass spectrometry, we know there's about 980 proteins in that area. So it's a finite number. It's a workable number.
You mentioned epilepsy in there. Is there a direct correlation with syndrome X, that there will be a direct correlation with epilepsy, or is that too?
So, well, about 20% of patients with Fragile X syndrome suffer from seizures.
Okay.
So it may be very helpful for those patients in particular. But generally, their seizures in Fragile X syndrome are controlled by already known seizure medications.
So with those controlled seizure medications, does it have any relationship to the outcomes of?
Not that anybody's noticed.
No.
Congressman mentioned a son is fully methylated. Now, is there a lesser number of repeats that would be like a pre-mutation? And what sort of symptoms are exhibited and how would you treat?
Yeah, so I don't want to get into the pre-mutations too much. Pre-mutation is kind of intermediate length of repeats, and those are found in carriers. And often, like the mother of a boy with Fragile X syndrome would be a pre-mutation carrier. Those repeats are unmethylated, so they make the FMR protein, so they don't have Fragile X syndrome, but they have other problems. So it turns out that men with the pre-mutation, particularly in their 50s and 60s and beyond, can get a tremor ataxia syndrome. So they get a neurodegeneration kind of phenomenon. And in women with the pre-mutation, about 25% of them suffer from premature ovarian failure. So these women can hit menopause quite early. And actually, I've met girls as young as 19 who've had menopause. So that has pretty significant consequences to those girls. But we're learning a little bit about those disorders too, but they're kind of different from Fragile X.
You mentioned the 1% of autism. What type of autism is that 1%?
So about one, well, actually, if you look at autism in general, you can break it up and say, well, generally, 3 or 4% is Fragile X syndrome. 1% is what we call 16p deletion, which is a little deletion on chromosome 16. Another 1% is on chromosome 15. So you kind of chop up these little bits of autism. So probably all told, maybe 10% of autism may have an identifiable origin. Those kids tend to be a little more severe than, so in the autism spectrum disorder, they're more towards the end where there's pretty significant intellectual disability. But you have 90% of autistic kids then that don't have any of those changes. So that's really the core group of autism that we have to look at.
You said you're not entirely certain why an increased number of repeats leads to methylation. Would there be any way to stop the methylation?
Yeah, so we've tried that in the past, and it's been difficult, but I think there's been recent advances in how we understand what things regulate gene expression. I think it's now a viable alternative again. And the problem is we haven't been able to study it because there's no animal model that recapitulates that expansion and turning the gene off. So the other thing I didn't mention, these are, as far as we know, still human-specific mutations. They don't occur in nature outside of humans. And we're unable to engineer a mouse to do that. So we haven't been able to study that step, but we can study it in stem cells. And so we've been unable, of course, to study that in this country. But in Israel, they've made stem cells from these patients, and they can show that methylation occurring in culture. So that now gives us a tool. And now, in the last year or so, there's been what's called induced pluripotent stem cells, which you can take from skin or hair and make stem cells without an embryo. And now we can do those kind of experiments. So I have 3 people in the lab doing just that. And the idea then would be to kind of get a system we can study that and maybe find a drug that might reverse that methylation.
My next question would then be, do you feel you'd have the same results as your antagonist?
Well, I mean, if we can turn the gene back on, I think it would be a much better situation because then that's eliminating the problem from the root cause. All these other drugs, really aren't going to recapitulate what normal is. But turning the gene back on could.
Has the flat NIH funding had any impact on the progress of your research or research in general?
Yeah, I think absolutely. I mean, I think the flattening of the budget has really, allowed fewer young scientists to get into the field, and I think that's the most critical part of the flat budget. Is we're not growing the new crop of investigators. Fortunately, research goes on outside the United States, but in many ways we are losing our advantages in leading the world in biomedical research because of the flattening. In Fragile X, I mean, there's been foundations that help. FRAXA, for example, funds a great deal of research, but of course they don't have enough money really to do everything. Simon's Foundation, Jim Simon's Foundation, he started not long ago, has funneled a lot of money into autism, and indirectly that's allowed work on Fragile X too. So, I mean, there are, but I think we need to really get back on track with the NIH.
Have your clinical trials been producing any preliminary data that you can share with us? Or?
No, that's the way the nature of these clinical trials are. You never know what's going on until the end.
There's no more questions. I want to thank you very much.
Thank you.